in our laboratory and in many others around the world, we're really keen to understand what is the underlying biology going on here, because if we can understand this, this can help us prevent these insults, help people to understand how important this time window is, and maybe develop interventions that will improve the prospects, particularly of pregnancies that are very vulnerable because of these sorts of insults, leading to preterm birth and preeclampsia. And what we find is that there is a pathway, and in fact several biological pathways interacting, by which many environmental factors that can impact the way an embryo and a fetus will develop are manifested in our biology, whereby our quality of nutrition, whether or not we have as parents, metabolic disorders, exposure to stress, infection, contact with lifestyle, drugs, smoking and alcohol. Increasingly, we're worried about environmental toxins and even manipulations around conception associated with assisted reproduction that can help and do influence the way an embryo will develop and will shape its future. And the pathways that are emerging to be most important are two pathways. The first one is the immune system, and the second one, and that's the immune system of the mother, and the second one is what we call epigenetics, and I'll tell you a bit more about those. So when you think about it, the immune system is really important in the reproductive process, and that's because an embryo gets half its genes from the father, it's got transplantation proteins on the surface of the cells as it develops, and theoretically should be rejected like a heart or kidney transplant is. So healthy pregnancy requires that the embryo develops a placenta and interacts with the mother's immune system in such a way as to not be rejected by that immune system. And for a long time we thought that was kind of because the immune system was closed off and kept away from the reproductive process. And indeed, that's wrong. The way that we now understand how these two systems are working together, the immune system that we always think is about defence and protection and rejection of foreign things, and the reproductive system where the tissues of the fetus and the embryo have to interact so closely with the mum, is indeed there is an immune response, but it's a special kind of immune response that allows the mother's body to tolerate these foreign tissues. Now, this is because the immune system has an incredible capacity to tell the difference between different kinds of challenges. There's the ones that are dangerous, like infections that we need to clear, tumours that we need to surveil and reject. So these are the bad guys. But then there's lots and lots of foreign material that comes into contact with our body every day that are harmless and in fact necessary for our biology to work properly. What we call the probiotic bacteria, the lactobacilli that we need to have our gut working properly, and the food molecules that are foreign that we ingest every day and have on our skin. So we have an immune response to these things, but it's what we call immune tolerance rather than immune rejection. So what happens in pregnancy is the same thing. The mother's immune system has to recognise the embryo as foreign, but then be convinced to mount immune tolerance, to accept that embryo as it first comes into contact with the surface of the uterus and begins to implant. So there are special immune cells that have to be present, and it's not important to know what they are, but they're the cells that mediate immune tolerance from right at the moment that embryo first makes contact. And the way these immune cells interact at that time of early implantation will then shape the way the placenta begins to develop and the way these placental cells invade into the uterus and access the mother's blood supply. And if the right immune cells are there, the blood vessels open up so that there's a strong blood supply to the placenta that allows a strong development of the fetus. Whereas if the immune cells aren't behaving properly, they'll either prevent implantation, lead to infertility, or close down these blood vessels so that they never support a strong, robust placenta. And that then has the impact on the way the fetal development is set in train. Now, the molecules that the immune cells are using to talk to each other are also talking to the embryo. And these molecules are called cytokines, and they're what we call signalling cells that are communicating between the immune cells and also between the mother's tissues and the embryo as it begins to develop and get ready to implant. So there's lots of good guy cytokines and also some not so good ones that can be secreted by maternal tissues to deliver this information to the embryo to shape it at this time. So all of those environmental factors that I told you about, the nutrition, the infection, exposure to environmental toxins, it looks like there are a lot of them operating through the immune system in the way these molecules are expressed to either support and encourage the embryo, or in some cases to not be so supportive and possibly even to cause damage and cell death in the embryo and expression of stress response genes. So the maternal support right from this early time, operating through the immune system, is really critical. But it's not just about the mum. It actually turns out that one of the key factors shaping this immune environment at conception is the father's tissues. And the seminal fluid delivered to the female tissues at the time of conception or in cycles before conception interacts with the immune system and delivers these molecules, and again, not important, but they're molecules that convince the female immune response to induce tolerance, prostaglandins, and a transforming growth factor beta molecule that we've discovered to be incredibly high in seminal fluid that drive immune tolerance. So the father's making an important contribution, and if we disrupt that contribution by doing mouse experiments while we let the mice mate and deliver either sperm and what we call the plasma fraction of the seminal fluid, or we have another group where we allow the mice to deliver sperm but not the plasma fraction because we've surgically removed the seminal vesicle glands, then we see quite remarkable effects in the offspring. First of all, we see changes in the embryo as they develop. So the embryos that are sired by fathers under normal circumstances are nice and strong and robust with lots of viable cells at the time they're getting ready to implant. But the embryos where the signals from the father haven't been delivered in the right way because of the surgical intervention to remove that plasma fraction, we can see now that the embryos are not being supported in the right way and they have less cells than they should at the time of implantation. And that's good evidence that shows that the paternal factors are contributing to this environment. What's really remarkable is when we look at the offspring. So when we compare, especially the sons of these two sets of fathers, we find that compared to the healthy dads that have healthy sons, the male mice that are born from the fathers where the seminal plasma wasn't allowed to instruct the female immune response correctly are now susceptible to metabolic disorder. So the embryos have been programmed in this thrifty way. They have all the hallmarks of metabolic disorder, showing how critical that conception environment is and the father's contribution as well. So the second pathway I'll tell you about briefly is epigenetics, and people will have heard about epigenetics. It's in the news, it's the latest fad, and it actually is really exciting biology that is instrumental in setting really the trajectory of early development and is interacting with the immune system to influence how these early embryos are developing. So epigenetics in a nutshell is really the extra information that's layered over the top of the genetics of the genes. And by these surface modifications to the DNA sequence, there's a variety of different epigenetic pathways by which the environment can shape the way the genes in a heritable fashion, so permanently in an individual, will or won't be expressed. And there are many pathways, what we call cytosine methylation, patterning of the chromatin, little non-coding snippets of nucleic acids called microRNAs, or the mitochondria in the cell as well. All of these are mechanisms by which the environment can permanently alter the epigenetic patterning. So in females, and again, this is much easier to do in animals, obviously, than humans, it's been shown in several studies that especially the oocyte, is very vulnerable to epigenetic programming, but also after fertilisation, the embryo, as it moves along the oviduct, undergoes these first cell divisions and gets ready to implant. Very vulnerable to reprogramming of the epigenetics in the maternal tissues. And an example of that here is in these mice that have a genetic disposition to make them eat too much and get too fat. And what we find in these hyperphagic blobby mice, they're called, is that their offspring are just as likely to be fat, but that information is carried through the mother's egg. And you can see the fat in the eggs even prior to the time they're fertilised, and already the fetus is developing differently as a consequence of that. And that is also linked with changes in microRNA and mitochondria. Remarkably and somewhat concerningly, there are many environmental chemicals that affect the epigenome, and this is an agricultural fungicide, vincosilin, shown in animal studies to impact, when delivered to the mums, the offspring, perhaps not surprisingly, but as we've heard from Ruth, because of the next generation also developing, there are transgenerational impacts right through to sons, grandsons and great-grandsons of a single exposure in a pregnant rat. But it's not just about the mums and her exposures, the fathers are important too. And paternal effects can be delivered, we now know, directly into the developing embryo at the time of fertilisation when the sperm fuses with the egg. This sperm is carrying DNA, but it's also carrying these epigenetic messages, the non-coding microRNAs. And depending on which microRNAs are carried, different information can go through about the father's environmental exposures. So a big one is smoking. Smoking damages DNA, we've known that for a long time, but also changes the patterns of these microRNAs. And that is potentially contributing to the explanation for the health consequences for sons of dads who smoke. It's not just smoking, poor diet, obesity, and exposure to environmental toxins, also altering the pattern of the epigenetics in the sperm to permanently change the epigenetics in the offspring. And what's really emerging, and I'm just going to show you one experiment that is incredibly fascinating, that is that there may be some specificity to this interaction whereby particular messages of epigenome carry particular information about prior experiences of the dads. And this experiment that's published in a very reputable high-impact journal, so it's really good science, showed that when male rats are conditioned to become fearful when they smell this organic chemical acetophenone, so they're conditioned to become fearful when they have that smell, their offspring also have the memory of that exposure, even though they've never been exposed to it. So the first time they're exposed to it, the offspring, they show an elevated fear response. Now that's incredible. You'd be thinking maybe the dads have somehow taught the sons for this to happen. Well, what was actually proven by doing IVF, taking sperm from the rats at one end of the university to the other end, transferring the sperm into the eggs and then studying the offspring, is that there never had to be any physical contact between the fathers, the mothers and the offspring. It was all carried in the sperm. And remarkably, there was a particular single microRNA in the sperm that seemed to carry that information. So this is very new science, and I think just one example, and obviously nothing like this yet in humans, but it does raise this incredible question of whether our biology has so much more power than we might have imagined. So this tells us that, I guess, our thinking about the way sperm and eggs contribute to shape us, very important, information coming through the gametes, extra information through the mother's tissues and especially the immune system, to shape the embryo and determine the way it will grow into a fetus in such a way as to impact the offspring. But also that we're uncovering now new pathways by which these various tissues interact, and especially the immune response and the epigenetic pathways are emerging is important. Last 2 slides. So what does this mean for us as individuals and for society? I think for people considering becoming parents, we have to really get the message out there that to maximise not just their fertility and their pregnancy health, but importantly the health of their children, there's a bunch of things they can do to make sure that their environment is as healthy as it can be. Avoid smoking and alcohol, eat top quality diets rich in vegetables, protein and micronutrients, maintain healthy weight and exercise. Increasingly, we're worried about environmental toxins, particularly plasticizers and other pollutants in the environment, so avoiding those and limiting stress. But as a community, as a society, as a government, as policymakers, there are also messages here. And that is that whenever we consider things we do to our environment and the way we build our social structures and our built environment, we have to think about the impact on future generations. So promoting awareness of, I think these are things we can do as a community, promoting awareness of having healthy exposures in couples even before they think about becoming pregnant as much as we can. That requires better decision-making around people's reproductive health and really educating children and teenagers that reproductive health is a gift and a priority for future generations, and policies that take into account when we're considering how to shape our cities, our environment, that consider the impact on the next generation, really important. And altogether this has given us this new sense, I think, that to be a parent is an incredibly responsible thing, but it starts very early, and both mums and dads are important, very important. And these are the people who support my work, especially the University of Adelaide, the Robinson Institute where I work, and the NHMRC that supports and funds the research. And if you're interested to find more information, you can go to the Your Fertility website here, made in Melbourne in Australia, that we all contribute to, the reproductive biology people in the country, and this also excellent website from Europe, Begin Before Birth. Thank you.
Thank you very much, Sarah. Now I'm going to ask Sarah to remain here and for Ruth to join us, and we have 5 minutes or so for any questions that you may have, and we have a roving microphone. So are there any questions of either speaker? Yes, down here. Yes, sorry. Oh, okay.
Thank you for a fascinating lecture, really interesting. And I had no idea that so many problems could happen early on. So my question is about epigenetics. And it's been my understanding that at the very early stages, the sperm immediately loses its epigenetic information. The DNA methylation essentially disappears, and the female a bit slower. But only a few divisions along, it starts to be recovered again, and then the process of differentiation starts. So my question is, where does the information get stored? Where and how does it get stored, the stuff that's taken off the sperm and the egg? And also, is there scope for these terrible consequences to happen at such an early stage? I know you're thinking about those kind of possibilities.
Absolutely, and that's a very insightful question. The biology, and Ruth may want to comment on this because she's an expert too, around the way the epigenome is wiped clean and then reapplied is still emerging. I think indeed there is information now that it never completely is wiped clean, especially from the female gamete. There is some residual information that stays there. But you're quite right that that sort of removal largely of the parental information, but then the reapplication of that information makes that an incredibly vulnerable window. So the real question is, how does this environmental information come from the external environment, our diet, exposure to air pollution, etc., to change the way that epigenome is laid down in that new pre-implantation embryo? And that's where I think the growth factors and cytokines might be part of the solution, but there'll be other pathways as well that we're yet to understand. And the sperm, the carriage of that sperm information, it does particularly seem to be these short non-coding pieces of DNA, the microRNAs, and they're never actually attached to the DNA, they're carried in another part of the sperm. So when they're delivered to the egg, they're persisting in the nucleus of the egg and available to interact with that new genome and to sort of then lay down their information. And somehow that information stays with every cell of that embryo through all of the cell divisions going forward. So it's such a vulnerable and important time. Do you want to add?
No, I don't think so. I think that's...
Okay, do we have any further questions? Yes, one over here.
Is it? Oh, yeah, okay, I'm live. I was just wondering, so the effects of the Dutch hunger winter in terms of the subsequent effects on metabolic health seem to correlate with very, very early events, conception, very early embryonic development. And we also have evidence, as you said previously, that effects such as preterm birth similarly seem to affect our risk of developing metabolic diseases later in life. So one event refers to very, very early events, and one that happens relatively later on during embryonic development. Are the underlying processes related to one another? Are they evidence of there just being more than one way to achieve the same ultimate outcome? Are you interested in there being a connection between the two at all?
So there's a lot of complexity there, and again, that's a pretty smart question. So a lot of preterm birth, and particularly preeclampsia, is caused by problems with the placenta that actually do originate very early on as the embryo first implants and begins to form those blood vessel connections. So in that case, the time frame for the application of the insult, if you like, is the same, and the biological pathways can be, reasonably thought to be similar. However, not all preterm birth is to do with things that went wrong in the placenta early on. There's also preterm birth that comes because of, an accident or a hypoxic stress or other things that can happen through completely disconnected from early pregnancy. And there's also problems like to do with infection, I guess, that can begin in later gestation. Now, often those sorts of injuries and insults are linked with inflammation and changing the immune system. And it seems there that the immune system and the inflammation is also a connecting pathway. So if the placenta gets an inflammatory insult later, then it isn't going to work so well, and that inflammation, or the consequence of that, can be carried through to the fetus. But in some ways it seems to be, I guess it's difficult to make the judgment, but different things I think are happening at different stages.
And we have a further question.
Hi, thanks very much. I have a question regarding symmetry. It's very easy, I think, to tell the difference between top, bottom, front, back. But how does a heart cell know what's left and right?
Okay, so essentially the downstream processes are still the same. Once a series of events have occurred, then it sets up a chain, a molecular cascade on only one side of the embryo. And so, in effect, that does set up a difference which perpetuates itself between the left and right of the embryo. But the really hard thing to understand is to work backwards and try and understand what the initial symmetry breaking event is. And at the moment, our best understanding suggests that it involves interactions between both the signaling proteins that I talked about and some biophysical properties of the embryo. And it seems that there are particular organelles on the surface of cells, and actually almost every single one of our cells have them. But at early in embryonic development, there's a group of cells that can sense, that are mechanosensing, so they can sense flow. And we think that what happens is that extracellular fluid is swept in just one direction across a particular region of the embryo, and that fluid flow is being sensed by these mechanosensors. And those mechanosensors, rather than these chemical messengers that I was talking about, these mechanosensors can then set up this cascade of events inside the cell that change gene expression. And because the fluid is flowing only in one direction across the embryo, then cells only on one side of the embryo sense that flow, and therefore they're the only cells that set up these new gene transcription cascades that will tell the embryo, tell cells of the embryo whether they're on the left or the right of the embryo.
Although we're a little over time, I think I've seen 2 more hands, so we'll be happy to take your questions. Thank you.
Many years ago, I had some exposure to some of this technology, long since retired, and there was efforts back then. I mean, doing the research is quite difficult on humans and so forth, and there was a lot of development going on about 10 or 15 years about taking human nuclear DNA and putting it into the cells of other animals, such as cows, which raises also, and creating blastocysts, which they could then experiment with. I'm just wondering, what's the current state of affairs with that type of research, given all the complexities you have in trying to figure out how the cell structures work?
I saw a tweet the other day about a hybrid between a rhesus monkey and a human. There's some kind of meme, you know, a rumour about something that happened 100 years ago, and I think it's garbage. I think it would be incredibly difficult to overcome all of the barriers that an embryo has to go through, all the quality control checkpoints that occur in normal development to ensure that every aspect of development is according to the clock. And the support that's required from all the other cell lineages and you know, these immune factors are very often quite species specific, so it's very difficult to see how introducing DNA from another species would overcome that interspecies barrier. There are also very specific lock and key mechanisms on the surface of the cell. But I guess if you override all of those barriers, looking at the literature when I heard this meme was, you know, I guess there are some species like, you know, the donkeys and horses where you can achieve an interspecies chimera. But that absolutely requires very, very close species connection to overcome these barriers. Okay. So I think the example you gave would never develop.
Was there one more question? No. I think it's quite time we wrapped up. Oh, there was one more? Oh, sorry, I didn't see you up there. I don't know that there's a microphone up there, but, oh, there is? In the centre, yes. And this is the last question, everybody.
Hello. Okay. You've talked about epigenetics as mostly leading to problems, I guess, in some ways, and that you know, the hyper efficiency of the hunger winter children. But to me, and also the other idea that's popped in my head as well is that, you know, if for example, if I'm a monitor or a goanna eating a cane toad, and I've learnt to fear, you know, the smell of a cane toad, and I've survived and produced children, so to speak, that maybe there's a healthy or a positive side to the epigenetics and the information that's passed on. And for example, in the famine condition, like if that famine had persisted for a number of years, like I'm imagining those children might have been better off. So could you make some comments about that?
Yeah, I think that's, again, very insightful, that there must be evolutionary drivers behind these processes, and absolutely where it's useful for the offspring to adjust the phenotype to match the environment, that's a good thing that brings biological benefit, fitness, and would be an evolutionary driver. It's where the insult doesn't reflect what's going on in the environment that there's a problem. So when you've got a bunch of people being exposed to effectively nutrient deficiency because they're eating McDonald's every night, and then having children who think they're being born into an environment of calorie deficiency, you know, that have this thrifty phenotype. That's where the problem lies, where there's a mismatch. And I actually think the example I meant to say that of the exposure of the male rats to the aromatic chemical and the fear response does provide proof of concept that specific memories of challenges that could be to the detriment of survival, if they can be carried forward into offspring, what an amazing pathway is that? And it does tell us that getting the epigenetics right is an incredible gift that parents, especially dads, can give to children by taking care of the epigenetics and making sure that the signals match the environment.
Well, I think with those wise words, we should thank both Sarah and Ruth for joining us tonight to impart their knowledge so clearly and succinctly. I have no doubt that you've all found it as fascinating and enlightening. So I'd like you just to show your thanks to both of you. And we would all like particularly to thank you, our audience, for joining us here at the Shine Dome and online. Tonight's talk will be made available on the Academy's YouTube channel shortly. And finally, we really do want to thank our series sponsors here, Jarrah Wines at Geostation and Edge Catering. Without them, you wouldn't have had the very pleasant start to the evening that you had. So thank you very much for coming. We look forward to seeing you here back at the Shine Zone in April for the next instalment of Science of Us. Good night.